Meghana. In this video, let's discuss about one of the topic from the research paper, that is deep learning approaches for classifying bacteria types using morphology of bacterial colony. So let's get started. So what is a bacterial colony morphology? A bacterial colony morphology refers to the observable characteristics of bacteria colonies as they grow on solid media. This includes various attributes such as shape, size, color, texture, and elevation. These morphological features are crucial for the preliminary identification and classification of bacterial species, as a different bacteria exhibit distinct colony characteristics. The overview of colony morphology is, the first thing is basically the shapes, and next one is size, color, texture, and elevation. Coming to the shape, colonies can be a circular, irregular, filamentous, or rhizoid. The size can be based on the diameter of the colony, which vary significantly from 1.2 pinpoint to several centimeters, and the colors may appear in the white, yellow, red, or other colors, often influenced by the type of the media that are being used, and the texture can be of smooth, rough, shiny, or dull. And elevation comes with the colonies can be a flat or raised, convex, or ubonate. What are the traditional methods versus the automated approaches that have been taken place? Traditional methods of bacterial classification often rely on visual inspection and manual techniques, which can be subjective and prone to human error. These methods are typically time consuming and require significant expertise, making them less efficient in high throughput scenarios. In contrast, automated approaches such as particularly those whereby utilizing the deep learning and offer a more efficient and objective means of classification. By analyzing the images of bacterial colonies, these systems can quickly and accurately identify the species based on morphological features without the need of extensive or any manual intervention. So basically, what are the problems that we are finding at, basically the challenges that we have to overcome is in the traditional methods that are basically, it is a time consuming. The process of culturing the bacteria and analyzing them can take several days, and also there will be delay in the diagnosis and also the treatment, where it requires expert knowledge in that. Accurate identification often necessitates a skilled microbiologist, which may not always be available for them. And the next one is limited accuracy due to the human error, where subjective interpretation of morphological characteristics can lead to inconsistency and also some misidentifications of the problem. So in order to overcome those problems, we are implementing a technique that is called as deep learning, which is automated analysis. Deep learning algorithms can process large data set of the bacterial images, significantly reduces the time and also that are required for the classifications, and also it is faster and scalable. These systems can be scaled to handle numerous samples simultaneously, making them suitable for the high throughput for the environments. Can potentially improve the accuracy by minimizing the human involvement. Deep learning approaches can enhance classification accuracy and reliability, leading to better outcomes in clinical and research settings. where this frame sets the stage for the exploring the application of deep learning in bacterial classification as discussed in the subsequent section of the study. So how this is implemented, basically the first and the fundamental step is basic in order to collect the data set. Like the dataset is used in the study was collected from the Hanoi Medical University in Vietnam. It consists of sRGB images of bacterial colonies taken from petri dishes with a pixel size of 3000 into 3000. The dataset includes images of three bacterial species. The first one is Escherichia coli, which is of 14 images, and Staphylococcus aureus, it is of 15 images, and the last one is Klebsiella pneumonia, it's of 11 images, where the images were carefully selected to represent the morphological diversity of the colonies. In order to enhance the dataset, augmentation technique were also applied on it, which include cropping, resizing, and generating an additional or an improved way in order to increase the robustness of the model, which is nothing but data pre-processing. Next is basically the model architecture, how we are planning or implementing the model. The deep learning model architecture employed in the study consists of several fine-tuned convolutional neural network, which is a part of neural networks that were pre-trained on the ImageNet dataset. The following architectures were investigated are VGG16, ResNet 50, MobileNet V1, and DSCNN, Deep Bacteria. Each model was selected based on its ability to capture complex features in the images and the architecture by optimized for the specific task of the bacterial classification. This model were also designed in order to process the input from any kind of thing, but with a size of uh, 2 the size of 224 into 224 into 3 and 227 into 227 into 3, which allowing for the effective feature extraction and also for the classification purpose. Next one is basically the model have been implemented. Like for that model we need to train and test the data sets. Like through the pipeline involved in several cases, the first step is basically data pre-processing. There are images which have been cropped to extract regions of interest and resized to fit the model input requirements, and the augmented to increase the dataset size. Dataset splitting, the processed images were divided into training and testing sets using a like suitable ratio to ensure a balanced evaluation. Model training, like the selected CNN architecture were trained on the augmented dataset with the model training in order to classify the bacterial species based on the morphological features of the colonies. The next one is model evaluation. After training, the model were tested on the unseen tests such as at the set of evaluate their classification performance with metrics such as an accuracy, precision, recall, and F1 score being calculated. Then finally, the results and the evaluation were based on the studies demonstrated that the deep learning models achieved high classification accuracy, and the best performing model reached an accuracy of 97.19% in classifying the three bacterial species. The evaluation metrics indicates that the model not only performed well in terms of overall accuracy but also showed a strong performance in identifying each bacterial species individually. The use of data augmentation and fine tuning of pre-trained models contributed significantly to the improved performance. So I can conclude that the study successfully demonstrated the effectiveness of the deep learning approaches in classifying bacterial species based on the colony morphology. By leveraging advanced CNN architecture and a well-curated dataset, the proposed system achieved high accuracy and efficiency in bacterial identification. The automated approaches, such as the potential to revolutionize bacterial classification in various fields, including clinical diagnostics and food safety, by providing rapid and reliable results. For further, like for future work, may focus on expanding the dataset, exploring additional architecture, and integrating the system into a practical applications for the real-time bacterial identification. Thank you for watching the video.